Welcome back to the deep dive. If you've ever learned biology, you've definitely heard the phrase, the mitochondrion is the powerhouse of the cell.
Right, that old chestnut.
Exactly. Well, today we are taking that simple description and honestly throwing it out the window. We're diving deep into the ultimate cellular machine.
Uh-huh.
And not just as a power generator. We're looking at it as like the central financial officer, the quality control manager, and really the ultimate decision maker for whether a cell lives or dies.
Yeah, it's really necessary to move past that simple textbook definition because the mitochondrion is, well, arguably it's the most spectacular evolutionary anomaly in biology.
Anomaly, how so?
Well, our sources really highlight this. It isn't just another part of the cell, another organelle. It's basically an ancient self-replicating organism, a prokaryote, like a bacterium that struck this incredible deal with primitive eukaryotic cells maybe, over 1.5 billion years ago.
Right.
And this partnership, this endosymbiotic union, that's really what allowed complex life like us to even flourish.
Okay, so that idea of the biological dual citizenship, you could call it, that's kind of the fundamental hook here.
Exactly.
So our mission today is to take this stack of sources we have, all detailing cellular metabolism, and really pull out the key insights. We're focusing on 3 core operational roles this amazing organelle plays.
Mm-hmm.
We want to get beyond just making energy to understand how its structure, its metabolic flexibility, and its signaling pathways literally govern whether a cell survives, divides, or undergoes programmed death.
And this isn't just abstract biology, right? This knowledge sits right at the intersection of things like aging, athletic performance, and major diseases. Think cancer, neurodegeneration. It's central.
Absolutely.
And if we really boil down the functions detailed in our research, we can kind of assign 3 core operational hats to the mitochondrion.
Okay, let's hear them.
First, it's the energy generator. That's the classic role, mainly through oxidative phosphorylation, super efficient.
Right.
Second, it's the metabolic chef. It's flexible. It prepares the raw materials, the substrates needed for growth, for building new cell parts.
The chef. I like that.
And third, and this is maybe the most complex role, it's the cellular judge. It holds the switch, basically, that determines cell survival or triggers programmed death, apoptosis.
Yeah. Energy generator, metabolic chef, cellular judge. Okay, that's a great framework. Let's unpack this, starting right at the foundation. I mean, for a machine to work this well, its architecture, its physical structure must be incredibly precise.
Oh, absolutely.
So let's look at that structure first. How does its design enable this incredible level of energy transfer and control?
Right. So when we look at the mitochondrial structure, it's immediately obvious that function dictated the form. It's not just a random blob. We're dealing with 2 highly specialized membranes.
2 membranes, okay.
Yeah, and these create distinct compartments inside. This separation is absolutely necessary for generating that powerful electrochemical gradient we'll talk more about.
Okay, let's start with the outside layer then, the outer membrane. What's its role?
So the outer membrane is kind of like the first boundary, and it's actually relatively permeable, surprisingly so.
Permeable.
Yeah, our sources note it's studded with these specific channel proteins called porins. Often they form what are called voltage-dependent anion channels, VDACs.
Okay.
Think of these channels like large general doorways. They allow pretty free passage for small molecules, anything under about 5,000 Daltons, roughly. So basic fuels, waste products, small ions, they can move relatively easily in and out of the space between the 2 membranes. It's like an initial filter.
Got it. So the outer membrane is porous, like maybe the outer wall of a city, letting small things through easily.
Here.
But the real control, the specialized activity, that must be happening at the second barrier.
Precisely. The inner membrane is where the real action and, frankly, the bottleneck happens. Unlike the outer one, it's highly impermeable. It tightly controls almost everything that passes through it.
Impermeable, okay.
But its most striking feature is its shape, its topology. It's not smooth at all. It's intricately folded into these deep, winding structures called cristae.
Cristae. Right. And what's the point of all those folds? Just looks complicated.
No, it's all about surface area. It's pure real estate. The inner membrane, thanks to these cristae, can have something like 5 to 10 times the surface area of the outer membrane.
Wow, that much.
Yeah, it's a huge increase. And that massive surface area is absolutely crucial because that's where the entire machinery of the respiratory chain is embedded. All those protein complexes needed for electron transport, for making ATP, you need that space to pack them all in efficiently.
Okay, that makes sense. More workspace for the energy factories. And these 2 membranes, they create distinct spaces inside, right? You mentioned compartments. We have the core matrix right in the very center. What's happening in there?
The matrix is like the central processing hub. It's a high traffic area. This is where the initial fuels, the breakdown products from glucose, from fatty acids get processed further. It contains most of the enzymes for the tricarboxylic acid cycle, the TCA cycle, sometimes called the Krebs cycle. We'll definitely get more into that. But essentially, the matrix takes the fuel, strips off its high-energy electrons, and loads those electrons onto special carrier molecules.
Got it. Fuel processing and electron harvesting in the matrix.
Yeah.
And then what about the space between the two membranes?
The inner membrane space.
Yeah.
Our sources really highlight how critical this compartment is. Remember, the outer membrane is porous, lets small things through, but the inner membrane is super tight, very impermeable.
Right.
So because of that difference, the intermembrane space ends up having a chemical environment very similar to the main cellular fluid, the cytoplasm, outside the mitochondrium.
Okay, so it's connected in a way to the rest of the cell. Why is that important?
It's vital, because this is the space into which those hydrogen ions, the protons, are actively pumped during respiration. This builds up that high concentration gradient we need for energy generation. It's also where key molecules like ATP, energy currency, and ADP, the discharge form, are rapidly swapped back and forth to keep the cell fueled.
That compartmentalization is amazing.
No.
Such a specialized structure.
Okay.
Now let's pivot to something you mentioned earlier, the genetics. This whole idea of it being an ancient bacterium, its genetic history must still be visible somehow.
Oh, it absolutely is. This is that genetic dual citizenship we talked about. Mitochondria have their own DNA. It's circular, just like bacterial DNA, and we call it mtDNA.
mtDNA.
Now, mtDNA itself only codes for about 1% of all the proteins in a cell. Seems tiny, right?
Yeah, sounds negligible.
But that tiny percentage is incredibly critical. It codes for roughly 20% of the proteins essential for oxidative phosphorylation, the very energy generation system we rely on.
Huh. So it kept the code for the really core power functions.
Exactly. And the sources also confirm that the machinery inside the mitochondrion for actually copying its DNA, reading the genes, transcription, and building proteins, translation, it operates much like a modern-day bacterium does.
How so?
Well, it uses different kinds of ribosomes than the rest of the cell. It even starts building its proteins with a specific molecule, N-formylmethionine, which is typical for bacteria. This actually makes mitochondria sensitive to certain antibiotics that target bacterial systems. It's a clear echo of their evolutionary past.
That's fascinating. But you said dual citizenship. Where does the cell's main nucleus come in?
Right, here's the complexity. While mitochondria have their own critical DNA, about 99% of all the proteins found in mitochondria, including most of the regulatory proteins, structural components, enzymes for other pathways, they're actually encoded by the DNA in the cell's nucleus.
So the nucleus is like the system administrator, controlling the overall infrastructure.
That's a great way to put it. The nucleus directs the building of most of the mitochondrial structure and regulates its function, while the mtDNA provides the blueprints for some irreplaceable core components of the energy machinery.
Okay, but wait. If the mitochondria themselves, the physical organelles, come almost entirely from the mother's egg cell during fertilization,
That's generally true.
then why isn't every single mitochondrial disorder passed down purely through the maternal line? This dual control must complicate things.
It massively complicates things, especially for diagnostics. You're right, the mtDNA itself is almost entirely maternally inherited. The sperm contributes its nucleus, but essentially none of its cytoplasm or organelles.
So you'd expect maternal inheritance.
You would, if the problem was always in the mtDNA. But because the functional mitochondrion requires proteins made from both the nuclear DNA and the mtDNA, a disorder can arise from a defect in either genome. So if the faulty protein happens to be one encoded by the nuclear DNA, the inheritance pattern won't be maternal. It could be autosomal dominant, autosomal recessive, or even X-linked, just following standard Mendelian genetics.
Wow. So you can't just look at the family history on the mother's side and know for sure.
Nope. You absolutely have to figure out which specific protein is defective and then determine whether that protein's gene lives in the nucleus or in the mitochondrion itself. It makes diagnosing mitochondrial diseases really challenging.
Okay, so we have this incredibly complex, dual-governed organelle, but it's not static, right? You mentioned it's dynamic. It's constantly changing shape, moving around.
Absolutely. You shouldn't think of mitochondria as just static, bean-shaped things floating around. They form this constantly shifting network within the cell. They're always engaging in these dynamic processes called fission and fusion.
Fission and fusion, like
Yes.
splitting and merging.
Exactly. Fusion is when two or more mitochondria merge together. Think of it like a networking mechanism. It allows them to mix their contents, share functional proteins, even empty DNA copies. This helps buffer against stress. If one mitochondrion is slightly damaged, merging with a healthy one can dilute the damage and share resources.
Okay, so fusion is like resource sharing, promoting the health of the whole network.
Correct. And fission, which is the splitting of a mitochondrion into two or more smaller ones, is just as important. It serves a couple of key purposes.
Like what?
Well, first, it's needed when the cell divides. You have to split up the mitochondrial network to make sure the new daughter cells each get some. But second, and this is really crucial for health, fission acts as a quality control mechanism.
Quality control. How does splitting help with that?
It helps to segregate or isolate damaged sections. If part of a mitochondrion becomes dysfunctional, maybe it's leaking reactive oxygen species, fission can pinch off that damaged segment, separating it from the healthier parts of the network. This basically flags the damaged piece for disposal.
Disposal.
Right.
Because these machines don't last forever. The sources estimate they have a remarkably short half-life, maybe just 1 to 10 days, depending on the tissue and how active it is.
Yeah, they're constantly being turned over.
So how does the cell identify and efficiently get rid of these damaged, potentially leaky organelles that fission has isolated?
It uses a specialized, highly targeted recycling system called mitophagy. Literally means mitochondria eating.
Mitophagy. Okay.
It's a specific type of a broader process called autophagy, which is like cellular self-eating or house cleaning. Mitophagy specifically targets old or dysfunctional mitochondria. There's a complex molecular pathway that recognizes the damaged organelle. Often it involves certain signaling molecules like PNK1 and parkin accumulating on the outer membrane of the stressed mitochondrion.
So there's like a molecular kick-me sign put on the worn-out part, a biological target for disposal tag.
Exactly that. Once it's tagged, the cell kicks off the formation of this massive double-layered membrane structure. It's called an autophagosome. It actually originates from parts of another organelle, the endoplasmic reticulum.
A double membrane.
Yeah, this autophagosome grows and expands until it completely engulfs the tagged mitochondrion, sealing it off entirely from the rest of the healthy cytoplasm. Think of it as putting the trash in a sealed bag.
Okay.
And then finally, the sealed disposal bag, the autophagosome containing the doomed mitochondrion, fuses with a lysosome.
Lysosomes, those are the cell's digestive centers, right?
Yeah.
Full of enzymes.
Precisely. The lysosome is like the cell's powerful digestive vat or recycling center. It fuses with the autophagosome, and its enzymes break down the old mitochondrion into basic components, amino acids, fatty acids that the cell can then reuse.
Wow. That's incredibly efficient.
It is. And this process, mitophagy, is absolutely non-negotiable for long-term cellular health. If mitophagy doesn't work properly, damaged, leaky, ROS-producing mitochondria start to accumulate. And that accumulation is a major hallmark of cellular aging and many age-related diseases, especially neurodegenerative conditions.
Okay, that detailed architecture and this constant renewal process, that's the foundation enabling its most famous job, energy generation. Let's move to that first hat you mentioned, the energy generator. The big process here is oxidative phosphorylation, OXPHOS.
Right.
Walk us through this high-efficiency process. How does it use oxygen to generate just massive amounts of ATP?
Okay, so OXPHOS is essentially the final main stage of energy extraction from our food. It takes the fuel, which by this point has been broken down, and it's energy captured in the form of high-energy electrons carried by special molecules like NADH and FADH2.
NADH and FADH2. Those come from the TCA cycle in the matrix, right? You mentioned that earlier.
Exactly. If you think back to the matrix, the TCA cycle's main job isn't really to make ATP directly. Its primary output is generating a huge volume of these electron carriers, NADH and FADH2. Think of them like specialized delivery trucks loaded up with high-energy electrons.
Okay, trucks full of energy potential.
Yeah. These trucks then travel to the inner mitochondrial membrane and deliver their cargo, the electrons, to the start of the respiratory chain.
And the respiratory chain is that series of protein complexes embedded in the inner membrane.
Precisely. It's a series of large protein complexes, usually numbered I through IV. The electrons get passed down this chain almost like a bucket brigade from one complex to the next.
Okay.
And each time an electron moves to the next step in the chain, it releases a little bit of energy. It's moving to a lower energy state. And the crucial thing is that released energy is immediately captured and used to do work.
What kind of work?
The work of pumping protons. Specifically, the energy from the electron flow powers these complexes to physically pump positively charged hydrogen ions, protons, H+ from the matrix, that inner core.
Across the impermeable inner membrane and out into that inner membrane space.
Ah, so this is how we build up that gradient you talked about. It's like actively pumping water uphill into a reservoir.
That is the perfect analogy. You're creating that hydroelectric dam. By pumping all those positively charged protons out into the inner membrane space, you establish two things simultaneously. A chemical gradient, way more protons outside than inside, and an electrical gradient. The outside becomes positively charged relative to the inside.
Right, because you're moving positive charges.
Exactly. This combined force, the chemical difference plus the electrical difference, is this massive potential energy source. It's often called the proton motive force. And those protons are now really, really driven to flow back down their gradient, back into the matrix, just like water wanting to rush down a dam.
Okay, so you've built up this huge potential. How does the cell actually harvest it to make ATP? It provides only one path back in. The inner membrane is otherwise totally impermeable to protons. The only way back into the matrix is through a very special enzyme complex called ATP synthase, sometimes called complex V.
ATP synthase. The name suggests its function.
It does, and it acts like a tiny, incredibly sophisticated molecular turbine. The proton motive force drives the protons to flow rapidly through a channel in the ATP synthase complex. This flow causes part of the enzyme to physically rotate, like a water wheel or a turbine spinning.
It actually spins.
It literally spins, and that mechanical rotational energy is directly coupled to a chemical reaction, sticking a phosphate group onto ADP, adenosine biphosphate, to create ATP, adenosine triphosphate. That's the energy currency the cell uses for almost everything.
Wow. So the proton flow spins the turbine, and the turbine generates ATP.
That's the essence of it. And this whole system, OXPHOS, is incredibly productive. As our source is needed, you typically get somewhere between 36 to 38 ATP molecules generated for every single molecule of glucose that goes through the whole process. Glycolysis, TCA cycle, and OXPHOS.
36 to 38 ATP per glucose, compared to just 2 ATP from glycolysis alone, right?
Exactly. It's vastly more efficient. That's why OXPHOS is the absolute bedrock for powering complex organisms and sustaining cellular activities over the long term.
Okay, that's maximum energy efficiency. But here's where it gets really interesting, showing the flexibility of this system. You mentioned the electron transport chain doesn't always have to be coupled or linked to making ATP.
Right.
The cell can choose a different path if its goal isn't energy storage, but maybe heat generation.
Precisely. This is the fascinating phenomenon of uncoupling. It's like deliberately introducing inefficiency into the system, but for a specific purpose. Non-shivering thermogenesis, making heat without muscle contraction.
How does it uncouple the system? Is there a switch?
There is. It involves a specific protein found in the inner mitochondrial membrane called thermogenin, which is also known as uncoupling protein 1 or UCP1.
UCP1. And where do we find this protein? Is it everywhere?
No, it's highly enriched in a specific type of fat tissue called brown adipose tissue, or brown fat. It's particularly abundant in infants and hibernating animals, but adults retain some too, especially around the neck and shoulders.
Brown fat.
Okay.
So how does UCP1 work?
When the body needs to generate heat quickly, maybe you're exposed to cold hormonal signals like norepinephrine activate UCP1 in the brown fat cells. UCP1 essentially acts as a regulated proton leak or an alternative channel across the inner membrane.
A leak. So instead of protons being forced to go through the ATP synthase turbine.
Exactly. UCP1 provides a shortcut. The protons, driven by that strong proton motive force, rush directly back into the matrix through the open UCP1 channel. They bypass ATP synthase altogether.
And if they bypass the turbine, no ATP is made. But the energy from the gradient has to go somewhere.
It's released directly as heat. The potential energy stored in the proton gradient is dissipated as thermal energy instead of being captured as chemical energy in ATP. The fuel, like fatty acids, is still being rapidly oxidized by the respiratory chain to maintain the gradient, but the energy output is switched from ATP to heat.
So it's like running the engine really fast, but disconnecting the drive shaft, just letting it generate heat. Metabolic burnout for warmth.
That's a great description. It sacrifices ATP yield for rapid heat production. It perfectly demonstrates that the mitochondrion isn't rigidly locked into just making ATP. Its output can be dynamically modulated based on the cell's or the organism's immediate needs.
Incredible flexibility. Okay, now for the unavoidable downside of using oxygen so intensively, the trade-off. Our sources highlight that mitochondria are a major internal source of reactive oxygen species, or ROS.
Yeah, this seems to be an inevitable cost of doing business with oxygen in this high-energy electron transport system.
Mhm.
The respiratory chain is amazing, but it's not 100% perfect.
What goes wrong?
Sometimes, particularly at complexes I and III, an electron can accidentally leak out of the chain before reaching the final step, complex IV, where oxygen is normally reduced to water. This escaped electron can prematurely react directly with a nearby oxygen molecule.
And that creates
That generates superoxide radicals,
O2.
O, which can then be converted to hydrogen peroxide, H2O2, and other highly reactive molecules. These are collectively known as ROS. They're reactive because they have unpaired electrons or weak bonds, making them chemically eager to react with and potentially damage other molecules like DNA, lipids and membranes, and proteins.
Okay, so some ROS leakage is just normal.
Mm.
If that's a natural byproduct, how does the cell usually handle it?
Under normal physiological conditions, cells have robust antioxidant defense systems. They have enzymes like superoxide dismutase, SOD, catalase, and glutathione peroxidase that are specifically designed to quickly find and neutralize these RS molecules, converting them into harmless water.
So there's a cleanup crew constantly working.
Exactly. The problem arises when this balance is disrupted. If ROS production dramatically increases, maybe due to mitochondrial damage, toxins, inflammation, prolonged low oxygen hypoxia, or even just aging, which can make mitochondria less efficient, or if the antioxidant defenses become overwhelmed or depleted.
And the ROS levels build up.
Right. When ROS production significantly outstrips the cell's capacity to neutralize them, we enter a state called oxidative stress. And this chronic oxidative stress, this persistent damage from ROS, is now widely recognized as a key contributor to the aging process itself, and a major factor in the development and progression of many diseases, including cardiovascular disease, cancer, and especially neurodegenerative disorders.
A constant internal battle then. Okay, that really covers the energy generator role, its flexibility, and its inherent risks. Now let's switch to that second hat, the metabolic chef. This is where we see that fascinating paradox where a cell seems to willingly choose massive energy and efficiency for the sake of growth and proliferation. This brings us to the famous Warburg effect.
Right. So if a cell's main goal is just survival, maintaining itself, maybe performing a specialized function without dividing, then high-efficiency AUX-PHOS is absolutely the way to go. Maximize ATP yield from fuel.
Makes sense.
But if the cell's primary goal changes to rapid division, like in development, wound healing, immune cell activation, or unfortunately cancer, then its needs shift. It doesn't just need energy, ATP, it needs building materials, it needs biomass.
Biomass, you mean stuff to actually build the new cell?
Exactly. New lipids for the cell membranes, new amino acids for proteins, new nucleotides for copying the DNA and making RNA. It needs the physical carbon skeletons to construct these molecules.
And AUX-PHOS doesn't provide those.
Here's the problem with AUX-PHOS from a growth perspective.
Okay.
It's too efficient at extraction. It takes the carbon skeletons from glucose or fatty acids and basically burns them completely down to carbon dioxide, CO2, and water, H2O, to get the maximum electron energy. There are very few carbon leftovers available to be pulled out and used as building blocks.
Okay.
Yeah.
You can't build a second house if you use all your lumber just to fuel the fireplace in the first one.
That's a perfect analogy. You need to keep some of the raw materials aside. So rapidly dividing cells, both normal ones and especially many types of cancer cells, employ a different metabolic strategy to address this.
What do they do?
They dramatically increase their uptake of nutrients, particularly glucose and often the amino acid glutamine. And then, crucially, they shift their core metabolism towards what's known as aerobic glycolysis. This is the heart of the Warburg effect.
Aerobic glycolysis, meaning they perform glycolysis even when there's plenty of oxygen around. Because normally, glycolysis is followed by OX-PHOS if oxygen is present.
Precisely. Instead of sending the pyruvate, the end product of glycolysis, into the mitochondria for complete oxidation via the TCA cycle and AUX-PHOS, they largely convert pyruvate into lactate and secrete it, even in the presence of abundant oxygen. It looks like fermentation, but happens with oxygen available, hence aerobic glycolysis, or sometimes aerobic fermentation. But the energy cost. We just said glycolysis alone only yields a net of 2 ATP per glucose, compared to OXPHOS's 36-38. That's a massive sacrifice in energy efficiency. Why is that trade-off worth it for these cells?
Because the primary goal has shifted from maximizing ATP yield to maximizing the flux of carbon through metabolic pathways that allow intermediates to be siphoned off for biosynthesis.
Okay, explain that. How does incomplete breakdown help?
By running glycolysis at a very high rate but not funneling all the pyruvate into the mitochondria, the cell ensures that key intermediate molecules within the glycolysis pathway itself, like glucose 6-phosphate, fructose 6-phosphate, and just before the TCA cycle, are available in high concentrations.
And these intermediates can be pulled aside. Exactly. They get diverted or spun off into other biosynthetic pathways. For example, intermediates from glycolysis feed into the pentose phosphate pathway, which generates NADPH, reducing power needed for synthesis, and the 5 carbon sugars, ribose, essential for making new DNA and RNA. Other glycolytic intermediates provide the backbone for synthesizing amino acids like serine and glycine, and also the glycerol needed for making lipids and cell membranes.
Got it. So by fermenting glucose to lactate, they keep the early stages of glucose breakdown running fast, creating a pool of building blocks they can tap into.
That's the core idea. They sacrifice ATP efficiency per glucose molecule, but by consuming vastly more glucose, they get both sufficient ATP and the crucial carbon intermediates needed to build duplicate sets of everything required for a new daughter cell. Glutamine often serves as another key source of carbon and nitrogen for these building projects too.
So the mitochondrion, or at least the metabolic network it's part of, acts like that financial officer, again deciding whether to prioritize immediate energy cash flow, OXPHOS, or long-term structural investment and growth, Warburg effect.
It's a perfect summary of the trade-off. This metabolic reprogramming ability highlights just how central mitochondrial function, or the decision to bypass parts of it, is to determining cell behavior maintenance versus proliferation.
That flexibility is truly remarkable. Okay, which brings us smoothly to the third and perhaps most profound hat, the cellular judge.
The decider of life and death.
The idea that the mitochondrion isn't just doing metabolism, but is the central hub, the integration point for signals that determine whether the cell survives or undergoes programmed death.
Yeah, it's really positioned uniquely to do this. It sits at the crossroads of energy status, oxidative stress levels, and damage signals. It integrates all this incoming information complex internal signals about DNA damage, protein misfolding, lack of nutrients, plus external signals like toxins or instructions from the immune system, and then it generates the final, usually irreversible go or no-go signal for cell death.
Okay, our sources detail 2 main ways the mitochondrion can execute this death sentence. Let's start with the more messy one, necrosis, catastrophic failure.
Yeah, right. Necrosis is essentially the cell exploding. It's generally considered an uncontrolled pathological form of cell death. It's typically triggered by sudden, severe external injury, things like exposure to a harsh toxin, physical trauma, or a complete loss of blood supply and oxygen, which is called ischemia.
So a direct, overwhelming assault on the cell. How does the mitochondrion factor in?
This kind of severe damage often directly assaults the integrity of the mitochondrial membranes themselves. A key event described in the sources is the formation or opening of something called the mitochondrial permeability transition pore, or MPTP.
MPTP. Okay, is this in the inner or outer membrane?
It's thought to involve components of both membranes, forming a large non-selective channel that spans across them. Its opening is often triggered by things like massive calcium influx into the cell and high levels of oxidative stress, which are common during severe injury.
And when this MPTP opens?
Disaster for the mitochondrion. Because it's a large, non-selective pore, its opening causes the immediate collapse of that carefully maintained proton gradient across the inner membrane. Remember our hydroelectric dam, the MPTP is like dynamiting the dam wall.
Ah, gradient collapses, so.
So ATP synthesis instantly halts. The mitochondrion swells rapidly due to osmotic influx, the outer membrane often ruptures, and the whole cell, now starved of energy and structurally compromised, swells and bursts open, spilling its contents out into the surrounding tissue.
And that spillage causes problems, right?
Yes, it triggers inflammation and further tissue damage. Necrosis is generally a dirty death, causing collateral damage. It's uncontrolled cellular collapse initiated by mitochondrial failure via the MPTP.
Okay, so that's the uncontrolled explosion. Contrast that with the other pathway. Apoptosis, Krebs cell death, right? A controlled, deliberate cellular suicide.
Exactly. Apoptosis is a much more orderly, systematic process. It's like a carefully managed demolition rather than an explosion. And the mitochondrion plays the absolutely central role in initiating the most common pathway for apoptosis, the intrinsic pathway.
How does that work? What signals does the mitochondrion listen to for apoptosis?
It integrates those signals we mentioned, intrinsic signals, like detection of severe unrepairable DNA damage, overwhelming protein misfolding stress in the ER, or perhaps signals indicating lack of essential growth factors. It can also respond to extrinsic signals like instructions from immune cells, T cells, telling a virally infected cell or a precancerous cell to die.
So it gathers all this intel. What's the trigger point? What's the critical step it takes?
When the sum total of these pro-death signals reaches a critical threshold, overwhelming the pro-survival signals the cell also receives, the mitochondrion executes the key decision step: mitochondrial outer membrane permeabilization, or MOMP.
MOMP. Okay, you're emphasizing this is different from the MPTP in necrosis.
Absolutely critical distinction. MOMP is not the catastrophic opening of a huge non-selective pore like the MPTP. Instead, MOMP is a highly specific, regulated event that permeabilizes only the outer mitochondrial membrane, leaving the inner membrane and the proton gradient largely intact, at least initially.
Specific and regulated. How is it controlled?
It's primarily controlled by a family of proteins called the BCL2 family. This family has members that promote apoptosis, like Baxenbach, and members that inhibit it, like BCL2 itself, BCLXL. The fate of the cell hinges on the balance between these opposing factions at the mitochondrial outer membrane.
A molecular tug of war.
Pretty much. When the pro-death signals win out, they activate Baxenbach. These proteins then change their shape, insert into the outer membrane, and oligomerize, they cluster together to form specific pores or channels through that outer membrane.
Okay, so MOP is the formation of these specific Baxenbach pores in the outer membrane. What gets through?
Several proteins that are normally confined within the inner membrane space are released through these MOMP pores into the main cellular fluid, the cytoplasm. The most famous and critical one is cytochrome C.
Cytochrome C, but isn't that a vital part of the electron transport chain for energy production?
It absolutely is. Normally, it's loosely attached to the inner mitochondrial membrane, shuttling electrons. But when MMP occurs and cytochrome C leaks out into the cytoplasm, it undergoes a dramatic career change. It switches from being an energy metabolism worker to being a potent death signal.
How does it signal death?
Once in the cytoplasm, cytochrome C binds to another protein called a POF1, a apoptotic protease activating factor 1. This binding triggers a PEF1 to assemble into a large wheel-shaped protein complex called the apoptosome.
The apoptosome sounds ominous.
It is. The assembled apoptosome then acts as a platform to recruit and activate specific initiator enzymes called caspases, specifically caspase 9 in this intrinsic pathway.
Caspase is the executioner enzymes.
Exactly. Activated caspase 9 then turns on a cascade of downstream executioner caspases, like caspase 3 and 9F7. These executioner caspases are proteases, enzymes that chop up proteins. They go on a controlled rampage throughout the cell, systematically dismantling key cellular components. They chew up structural proteins, leading to cell shrinkage. They activate enzymes that chop up the DNA in the nucleus. They basically ensure the cell is neatly dismantled from the inside out.
So it's a clean, contained demolition.
Yes, the cell shrinks, breaks into small membrane-bound apoptotic bodies, which are then efficiently cleaned up by neighboring phagocytic cells, usually without causing inflammation. It's a tidy suicide.
So recapping the judge rule. Necrosis is mitochondrial collapse via MPTP leading to cell explosion, while apoptosis is a regulated decision leading to MOMP, cytochrome C release, apoptosome formation, and clean caspase-driven dismantling.
That's the core distinction. And understanding which pathway is being triggered and how it's regulated, particularly that MOMP step involving Baxenbach, is absolutely central to understanding and potentially treating many diseases. The mitochondrion, through its control over MOMP, is the gatekeeper.
Let's talk more about that clinical relevance then. How does the failure or dysregulation of these mitochondrial death pathways drive disease? Let's start with malignancy, with cancer.
Right. Cancer is, in many ways, fundamentally a disease of cells refusing to die when they should.
They evade apoptosis.
Exactly. Cancer cells typically accumulate a lot of DNA damage, they experience stress from rapid growth, and often poor blood supply conditions that should trigger the intrinsic apoptotic pathway via MOMP. But they very often find ways to evade this fate.
How do they do that?
One of the most common mechanisms is by messing with that BCL2 family balance. Many cancer cells dramatically overexpress the anti-apoptotic members like BCL2 or BCL-XL. These proteins effectively sit on the mitochondrial outer membrane and actively block back from forming those critical MMP pores.
So they jam the execution switch in the off position.
Precisely. Even if the mitochondrion receives strong internal signals telling it to initiate death, the overabundant anti-apoptotic proteins prevent cytochrome C release. The cell, despite being damaged or abnormal, survives and continues to proliferate. This failure of apoptosis, often combined with that Warburg metabolic switch we discussed, enabling rapid growth, creates a perfect storm for tumor development and resistance to therapy.
Okay, so cancer is often too little apoptosis. Now, flip that coin. What about neurodegeneration? Diseases like Alzheimer's, Parkinson's. Is the problem there too much apoptosis?
Yeah,
Overly sensitive, Judge?
that's a leading hypothesis for many neurodegenerative conditions. Unlike cancer cells, neurons are typically postmitotic. They don't divide. They're long-lived cells that rely heavily on efficient XPHOS for their high-energy demands. In diseases like Alzheimer's or Parkinson's, neurons are subjected to chronic stress, maybe from the accumulation of misfolded proteins, like amyloid beta or alpha-synuclein, increased oxidative stress associated with aging, or impaired mitochondrial function itself. This chronic stress seems to inappropriately lower the threshold for triggering MMP.
So they become too prone to undergo apoptosis.
It appears so. The apoptotic pathway gets activated prematurely or excessively, leading to the progressive death of neurons in specific brain regions. This neuronal loss is what underlies the cognitive decline in Alzheimer's or the movement disorders in Parkinson's. Here, the therapeutic challenge is the opposite of cancer. Can we find ways to inhibit inappropriate MMP, stabilize the mitochondrial outer membrane, and keep that apoptotic switch safely locked in the survival position for these vital, irreplaceable cells?
It's incredible how this one organelle, this ancient bacterial guest, sits at the very center, dictating cell fate growth, energy production, or death based on such a complex integration of signals.
It truly is the cell's command center in many respects.
Okay, so building on this foundational understanding, where is the research heading now? What are the cutting edges in trying to leverage this knowledge therapeutically?
Well, the detailed understanding of these pathways, metabolism, death signaling, quality control, has really opened up avenues for highly targeted research. These three areas are major frontiers right now.
All right, let's revisit the metabolic switch, the Warburg effect. If cancer cells are so reliant on this inefficient but biomass-producing metabolism, how are researchers trying to exploit that vulnerability?
Yeah, this is a huge area in cancer research. The idea is to hit cancer cells where their unique metabolic adaptation makes them vulnerable. Researchers are intensely focused on developing drugs that inhibit the specific enzymes crucial for maintaining that aerobic glycolysis or the related glutamine addiction.
Can you give an example?
Sure. One target is an enzyme called pyruvate kinase M2. PKM2. It exists in a form that's highly expressed in many tumors and seems to help regulate that balance between energy production and shunting intermediates towards biosynthesis. So, drugs targeting PKM2 aim to disrupt this balance. Another major target is glutaminase.
What does glutaminase do?
It's the first enzyme in processing glutamine, which, as we mentioned, many cancer cells gobble up almost as voraciously as glucose. They use its carbon and nitrogen for building blocks. So inhibiting glutaminase is an attempt to starve the cancer cell of this alternative fuel and building material source.
So the strategy isn't necessarily a blunt poison, but more like cutting off the specific supply lines that the Warburg-adapted cells rely on for construction materials.
Exactly. You're trying to metabolically reprogram the cancer cell, maybe force it back towards OXPHOS, which might slow its proliferation, or simply starve it of the precursors it needs to divide. It's a more nuanced approach, leveraging our understanding of that metabolic flexibility, or in cancer's case, metabolic rigidity in its adapted state.
Okay, that's targeting metabolism. What about targeting the death switch itself? That MOMP step the point of no return for apoptosis involving backs. How are researchers trying to precisely manipulate that?
This is another really active, though challenging, area. The goal is to find small molecules that can directly influence whether those back pores form or not. It's tricky because it involves controlling protein interactions and protein conformation right at the membrane surface.
What's the goal in cancer for manipulating MOP?
In oncology, the dream is to develop drugs, sometimes called BH3 mimetics, that mimic the action of the cell's own pro-apoptotic signals. These drugs would essentially bind to and inhibit the anti-apoptotic BCL2 proteins that are keeping Baxim back in check.
So unleashing Baxim.
Precisely. By neutralizing the inhibitors, these drugs would allow Baxim to oligomerize, form the MOMP pores, release cytochrome C, and trigger apoptosis. Even in cancer cells that have become resistant through overexpressing those anti-death proteins, it's about directly reactivating the suicide program.
And conversely, for neurodegeneration, you'd want the opposite drugs that prevent Bax from forming cores.
Exactly right. For conditions where excessive apoptosis is the problem, the search is on for compounds that could stabilize the outer membrane or somehow interfere with Bax back activation or oligomerization. You'd want something that acts like a supercharged anti-apoptotic protein, specifically protecting neurons from stress-induced MMP.
The challenge must be specificity, right? You don't want to shut down apoptosis everywhere in the body.
That's the huge challenge. Apoptosis is essential for normal tissue turnover, for eliminating infected cells, for immune function. So any therapy targeting MMP needs to be highly selective, either targeted specifically to the diseased cells, or perhaps addressing the specific upstream stress signal that's inappropriately triggering MMP in those cells. But the potential payoff is huge, because MMP is such a decisive control point.
Okay, metabolism, death control. What about the third area, quality control mitophagy? You mentioned its decline is linked to aging. How is research approaching the optimization of this cellular cleaning process?
Yeah, realizing the importance of mitophagy, especially in aging and age-related diseases, has spurred a lot of research into understanding exactly how it works and how it might be boosted. We need to know the molecular details. How does the cell reliably identify only the damaged mitochondria for destruction?
What pathways are involved in that tagging?
Key pathways are being intensely studied, maybe the most famous being the PINK1 Parkin pathway. PINK1 is a protein that accumulates specifically on the outer membrane of damaged mitochondria with low membrane potential. This recruits Parkin, an enzyme that then tags the mitochondrial surface proteins with ubiquitin chains.
Ubiquitin, the tag for destruction.
Exactly. These ubiquitin tags then serve as a signal recognized by the autophagy machinery, initiating the formation of that autophagosome to engulf the mitochondrion. So researchers are actively looking for ways to enhance this pathway or other related mitophagy pathways.
Enhance it how? Like finding drugs that boost PNK1 or Parkin activity?
Potentially, yes. Or finding ways to improve the recognition of the ubiquitin tags, or enhance the efficiency of autophagosome formation or fusion with lysosomes. The goal is to develop interventions, maybe pharmacological, maybe even lifestyle-based, like specific forms of exercise or dietary changes known to stimulate autophagy that could act as mitophagy enhancers.
Like giving the cell's cleaning crew a boost.
Precisely. Especially in tissues that are highly reliant on mitochondrial health and heavily affected by aging, like skeletal muscle and neurons. The thinking is that by improving mitochondrial quality control throughout life, we could reduce the accumulation of dysfunctional ROS-spewing mitochondria.
And that could slow down the damage cascade.
That's the hope. By proactively managing the health of the mitochondrial population, reducing the burden of chronic oxidative stress and inflammation they can cause when they fail, we might be able to mitigate some aspects of age-related functional decline and disease susceptibility. It's shifting focus from just dealing with damage after it happens to actively maintaining the quality of the cellular hardware.
Fascinating. Okay, let's try to wrap this up. This deep dive has really hammered home why just calling the mitochondrion the powerhouse, while true, is such an understatement.
It really is. It's this spectacular evolutionary relic, this former bacterium that now functions as, well, as we said, the cell's energy generator, its metabolic chef, and its Supreme Court judge, issuing these final verdicts on life and death.
And we've seen it operates based on these profound trade-offs. You've got the incredible efficiency of OX-PHOS for just pure energy maintenance versus the necessity of something like the Warburg effect to get the carbon building blocks needed for growth and proliferation.
Right. And underpinning all of that is its dynamic nature. It's not static. It's constantly undergoing fission and fusion, networking, and quality control checks, and being renewed through that indispensable cellular recycling process, mitophagy.
So boiling it down, what does this all really mean for you, the listener? I think understanding these mechanisms shows that cellular health isn't just a simple state of having enough energy. It's this continuous dynamic negotiation.
Yeah, a balance between maintaining structural integrity, those crucial membranes, achieving metabolic flexibility, switching between OX-PHOS and biomass production as needed, and having properly functioning programmed death signaling pathways. Health is about keeping all those systems functional and in balance.
Maintaining the integrity of those inner membranes, the efficiency of the transport chains, the precision of the signaling.
And the effectiveness of the cleanup crew, mitophagy.
Exactly. Which leads us perfectly into our final provocative thought for you to mull over. We've talked about how chronic diseases and the aging process itself might be accelerated by damaged mitochondria accumulating because mitophagy becomes slow or inefficient over time. So the question is, what preventative measures, if any, could a person potentially take right now thinking about nutrition, lifestyle, environment to actively support or enhance their body's natural ability to perform mitophagy? To help the cellular judge always have clean, functional machinery to work with, ensuring cellular house cleaning stays efficient.
It shifts the focus, doesn't it? From just passively accepting aging or decline to thinking about active lifelong maintenance of this critical cellular system.
Something to think about. Mull that over until our next deep dive.